Hello boys, welcome back to another session of video tutorials. This session is meant for class 11th biology students. This is meant for 3rd August 2020. Our memory always helps us to recollect and reflect upon what we have studied and understood. As we understand, we have studied in our previous session meiosis, the process of meiosis, and you may recollect that meiosis is the equal division, equal division, that is one parental cell divides and forms two equal daughter cells. When we say equal, in all respects the daughter cells are equal to parental cell. Like in the case of human beings, our own case, if one parental cell having 46 chromosomes, that's our chromosomal number, divides, it forms two daughter cells and each daughter cell will have 46 chromosomes. So the main significance of meiosis is maintaining the identity of the cell, of the tissue, of the organisms. Hence we say equal division. Whereas in today's session we are going to discuss and reflect upon meiosis. Unlike meiosis is a reduction division, RD. Mitosis, ED, equal division. Meiosis, RD, reduction division. Why reduction division? The daughter cells that are formed, unlike in meiosis, the chromosome number gets reduced. By how much it gets reduced? By 50%. That is the reason why we are using the word reduction division. So meiosis is equal division, meiosis is reduction division. That means normally we say a cell having chromosome numbers in doubles, like two sets, we say 2n, as I said. N is equal to number of sets. How many sets are there in a cell? Two sets. Then we say such a cell as diploid. Di means two. Ploidy, sets of chromosomes. How many sets are there? Two sets of chromosomes. So any cell that is having two sets of chromosomes is said to be diploid cell. When a diploid cell undergoes meiosis, first of all, we must understand that meiosis is a reduction division. What is getting reduced? 2n diploid is becoming haploid, means number of sets of chromosomes from n, to begin with it is 2n, it becomes n. Like if you take a human cell, a cell having, parental cell having 46 chromosomes, at the end of the meiosis will become 23 chromosomes. Like in human cell, as all of us know, 46 chromosomes. In 46, 44 are bodily chromosomes or autosomes, chromosomes that are responsible for deciding the characters of the body, bodily characteristics. And rest 2, 44 plus 2, those two are called allosomes or sex chromosomes. Normally they are X and Y. So presence or absence of Y will decide the sex of the baby. If Y chromosome is present, then it is male. If Y chromosome is absent, then it is female. So directly meiosis helps in deciding the number of chromosomes that are to be formed in daughter cells. And in the daughter cells, depending upon whether X chromosome is present or Y chromosome is present, almost it decides the sex of the baby. That is the significance of meiosis. So it happens in two broad phases. We say meiotic 1 or meiosis 1 and meiosis 2. What happens in meiosis 1? It's the reduction number. That is 2n has become n. And what happens in meiotic 2? N continues and forms equal division. That means meiotic 1 is RD, reduction division. Meiotic 2 is ED, equal division. We can say meiosis 2 is almost mitosis. So at the end of the meiosis, what happens? As you are seeing, from one diploid parental cell, we are getting 4 haploid daughter cells. This is number 1. And second, in mitosis only 2 daughter cells. Here in meiosis 4 daughter cells. So what happens in meiotic 1? The diploidicity is maintained. Homologous chromosomes get separated. What do you mean by homologous chromosomes? Already we have said chromosomes that look alike, that look similar in all respects are called homologous chromosomes. So the homologous chromosomes get separated. In meiotic 2, now chromatids are getting separated. What are chromatids? The thread-like structures. So in meiosis 1, chromosomes are getting separated. In meiosis 2, chromatids are getting separated. This is what you must remember. So what happens in meiosis 1? So it is divided into 4 stages: prophase 1, metaphase 1, anaphase 1, telophase 1. And prophase 1 is the longest period consuming phase here. The chromosome number, you can see here in prophase number 1, nuclear membrane is clear, nucleus is clear, chromosomes are short and thick, and in late prophase also we can see the formation of spindle fibers. And in metaphase, nuclear membrane disappeared, nucleolus disappeared, chromosomes are in the center of the plate. That's what I said. How to recognize whether cell is in metaphase 1? It is that way. If the chromosomes are in the center of the plate, that's metaphase. And in anaphase, you can see all along the spindle fibers, the chromosomes are moving north pole and south pole, opposite poles they are moving. That means when 46 chromosomes are there, 23 are going up, 23 are down. And then in telophase 1, cytokinesis takes place, cell division takes place. Cell gets constricted and cytoplasmic wall will be constructed. 2 nuclei will be formed. Nuclear membrane reappears, nucleolus reappears, and spindle fibers disappear. Finally, you get 2 daughter cells with n number of chromosomes. That's why we say meiosis 1 is reduction division. So these are the broad characteristics of prophase 1. You can go through it, read that, and then in prophase 1 you can see the most important one, homologous chromosomes. They form tetrad, and at some places they cross over. Wherever they are cross over, that crossover area is called synapsis. Now you can see here crossing over, and this is most important in meiosis. Crossing over of chromatids will make exchange of genes, as you are seeing. You can see here yellow chromosome with the red chromosome, and the pairing of homologous chromosomes, and then they cross over each other. And then look at the third diagram here. You can see exchange of portion of chromatids. When they exchange portion of chromatids, that portion of genes also get exchanged, and that's how the new varieties of species will be formed. Variations will occur thanks to synapsis and crossing over. Now you can see here a lot of synapsis and crossing over. You can see further, crossing over portions. And in metaphase 1, as I said, all now we can say hybrid chromosomes. You can see the color of the chromosomes also in the center of the plate. In anaphase 1, movement of chromosomes. This is important. Movement of they are not now homologous. We can't say now they are heterologous chromosomes. And telophase 1, spindle disappears, constriction of wall, cytokinesis. Main thing is cytokinesis with the 2 nuclei. And in meiotic 2 or meiosis 2, only 1 homologous of each chromosome is present in the cell, because already their hybridization has taken place. And sister chromatids carry identical genetic information. So in meiosis 2, chromosome number gets reduced. Now here you can see in prophase 2, these parental cells now, you must remember, they are daughter cells of the meiotic 1. So 23 are there. So 46 has been reduced to 23 chromosomes. 2n is reduced to n. Now in metaphase 2, you can see that n number of chromosomes are lying in the center of the plate as usual. In anaphase, most important, what is the difference between anaphase 1 and anaphase 2? You must have the clarity. In Anaphase 1, it is the chromosomes that are moving, whereas in Anaphase 2, it is the chromatids that are moving. Okay, please keep remembering that. And telophase 2, cytokinesis takes place, and from one parental cell we are getting two, another parental cell we are getting two, totally four identical haploid cells. That's important. N number of cells are formed. So in prophase 2, nuclear envelope fragments, spindle formation starts. Metaphase 2, very, very characteristic. What is the difference between metaphase 1 and metaphase 2? Please identify it. Write down on a paper what are the differences. Similarly, anaphase 1 and anaphase 2, just now I said, sister chromatids separate and move to opposite poles thanks to nucleolus. And telophase 2, nuclear envelope assembles, chromosome decondenses, spindle disappears, cytokinesis divides cells into two. So 4 cells are formed. And how do they help? They help in the formation of egg and sperm. Just as I said, 4 haploid cells with a copy of each chromosome, 1 allele of each gene. Different combinations of alleles for different genes along the chromosome. This helps in gametogenesis, oogenesis as well as spermatogenesis. Already these concepts we have covered in chapter number 4. Hence, the significance of meiosis is it directly helps in the formation of